Welcome to this video, and there's quite a lot of scientific work now that's really very good news. There's a lot of evidence emerging that immunity to SARS coronavirus 2 is going to be long-lasting immunity. And this has got huge implications. It really means that we can develop herd immunity. It means the vaccines are more likely to work, and it means this disease could potentially be eradicated from the surface of the earth. And I'm going to look at some studies that give evidence that point strongly in that direction. Now, I've been fairly hopeful about this for some time now, but it's good to see that the evidence is now starting to come through. Good news on this video. But before we do that, I want to do something that's really irritating me. Where did this virus that's infecting and causing this cluster in New Zealand come from? Only briefly before we go on to the good news. Now, they've ruled out frozen food items or freight as of today, Tuesday. Last week they already ruled out burning ember, and they've ruled out infection from returners. So the burning ember would have been like someone who was asymptomatic passed it on to someone who was asymptomatic, and it's a residual. But they had 102 days without any community transmission, so that was always unlikely. But they've ruled those three things out now, so what does that leave? It's hard to see what it leaves. Migrating birds, zoonotic reservoir. Don't really know, but that's where we're at now. So they're ruling things out, but they haven't ruled in anything yet. Now, I assume they've got a full phylogenetic analysis of this virus, and they know which viruses around the world it's similar to. But if they have, if they do know that, which they probably do, they haven't yet released that information. Now, New Zealand itself, another 12 new cases on Tuesday today. So that was Monday, that was Sunday, that was Saturday, that was Friday. So modest number in new cases, 1,643 cumulative in New Zealand, and 6 hospitalised as a result of the current outbreaks, no deaths. But where it came from, fascinating. Will we ever find out? You know, maybe we'll never find out. We still don't know where the original virus in Wuhan came from. That's still being debated. There's no absolute science on that yet. Now, the next few minutes I'll give you the bottom line on this video if you want to skip the science. Now, it's obvious that long-term immunity would be good. Antibodies provide real-world protection would be good. Now, what we mean by real-world protection is, does it actually work against wild strains of the virus caught in the real world? So this is not a laboratory setting. This is very much an in-life, in-community, in-pandemic response. So we want the antibodies to provide this real-world protection. And if that's the case, that would be brilliant. We want cellular immunity that is long-lasting, the immune cells. We want them to be there for years. And these are the memory B and the T lymphocytes. And I will be going over this after I've given you the bottom line. These are the T helper, suppressor, killer, and the killers are the cytotoxic cells. If you want to know that, stick around, we'll do that in a minute. And we want this to be true even in milder cases, and of course we want people, even who've had asymptomatic disease, to be immune. So whether you've had severe disease, mild symptoms, or completely asymptomatic disease, ideally we would like that to confer immunity. So many people could become immune if this works out, with not even knowing they've had the disease. So all those things would be good if they happened. And it's starting to look like all of these ticks probably will be ticked off. This is starting to look encouraging. I'm going to give evidence today for all of these actually being true. There's just torrents of good news on today's video. It really is quite incredible. Now, antibody levels so far interesting. Now, these antibody levels only mean anything if antibodies confer immunity. And as I say, we now have evidence that they do. So now the antibody levels, these are based on serological community studies of how many people that have been exposed and developed antibodies. Let's go through them. I mean, the UK ones, for example, we know the antibodies might not last for more than 3 or 4 months. But we can add together all the people that have tested positive for antibodies at any one time and come up with a number that have been infected with the virus. And of course, this is moving towards herd immunity. Now, there's a big debate about herd immunity itself. Some people are saying 70%, some people are saying 50%. Actually, it's a gradated scale. I mean, if 40% of people were immune, that's going to slow down the spread of the virus. But if 50% of people were immune, that would slow it down even more. And if 60% were immune, that would slow it down even more. And if 70% were immune, that would probably mean that the virus would die out. So herd immunity is not sort of a single thing. The more people that are immune, of course, the better. Now, crowded areas in India, New Delhi and Mumbai, studies have shown 24% of people already got antibodies there. They've been exposed to the virus. A quarter of these massive populations have already been exposed. Moscow is probably 21.7%. Stockholm is about 10%, lower in the rest of Sweden, but higher in Stockholm. New York city area, 6.9%. London, 17.5%, really very high in London. And 6.2% for the UK altogether. So that's kind of where we're at. So obviously, in these areas where antibody levels are higher, there's going to be more immunity. Spread is going to be harder than if that were 7%, for example. It's going to give a degree of, I don't like to use the term herd immunity, but it's going to slow the spread of the virus down somewhat. Now, still on the bottom line, I'm going to tell you what some major boffins have been saying about this study I'm about to give you, and a couple of other studies as well. So this is Marion Pepper, immunologist, University of Washington, talking about this study I'm about to reveal unto you. It's exactly what we would hope for. All the pieces are there to have a totally protective immune response. Sounds good to me. Immunologist, University of California, Davis. In Davis, this is very promising. Calls for some optimism about herd immunity and potentially a vaccine. So important implications for herd immunity and immunity that will be induced by vaccination, or immunity that is very likely to be induced by vaccination. Professor Danny Altmann, Department of Immunology and Inflammation, Hammersmith Hospital, Imperial College London. While this is a small-scale study, it offers a remarkably real-life human experiment at a time we've been short of hardline formal proof that neutralizing antibodies genuine offer protection from reinfection. In short, it's good news. Right, so that's the bottom line. It's looking like the studies out now that are indicating that antibodies are effective and that immune cells are effective in preventing further cases of disease. Now, so that's the bottom line. So if you want to switch off now, that's fine. But do stick around for the interesting bit. Be there or be square. Because this is really the interesting bit comes now. So, immune cells. There's different sorts of immune cells, but there's one type called lymphocytes. So the lymphocytes are the immune cells that primarily work against viral infections. And there's two sorts of lymphocytes. And technically, they're known as big or small. So that's not too hard. Now, the big lymphocytes are the natural killer cells. The cells that will go around killing other cells that are infected with viruses. But these work in a fairly non-specific way. So these natural killer cells, if they see viral residue on the surface of a cell, they'll just go there and kill it. So what happens is when a cell is invaded by a virus, the cell will sort of smuggle some of these viral particles onto the surface of the cell, thereby warning the immune system that that cell is virally infected. Then the natural killer cell, if it recognises virtually any viral residue, will just kill that cell, thereby killing all the viral particles inside of it, therefore preventing their reproduction. Causes quite a lot of inflammation. That's why when you've got a sore throat it goes red. It's not the virus that's making it go red, it's these natural killer cells killing all the cells that are virally infected. It's the body's inflammatory reaction that's making it making it painful for you. So now the small lymphocytes, they're divided into B and into T. Now these are so called because the B lymphocytes mature in the bone and the T lymphocytes mature in the thymus gland. Now the B cells differentiate into what's called plasma cells. And these plasma cells are the actual factories that produce the antibodies. The immunoglobulins. Antibodies and immunoglobulins are the same thing. The immune proteins that kill the viral infection. And these are specific to a particular type of virus. Now, the T lymphocytes, slightly more complicated. There's T helpers, there's T suppressors, and there's T killers or T cytotoxics. So those three types of T cells. Now these all work in a specific way to a particular virus. So what happens is that the T helper cells actually stimulate the B cells and the plasma cells to produce the antibodies. Now it's the T helper cells that are infected and killed in HIV. So in HIV, the reason someone is immunosuppressed is because the helper cells fail to stimulate the B cells because they've been killed. Then when there's enough immune response, the suppressor cells kick in and reduce it. But the cytotoxic T cells, just like their big brothers, the natural killer cells, but these work in a much more specific way. So what these T cells will do, so that's your cytotoxic T cell there. What it will do, it will come to a cell like that, a body cell, that's one of your body cells. And if this body cell is infected with the virus, a specific virus in the case of the T cells, in this case, of course, the SARS coronavirus 2 virus, if it recognizes the presence of these viral residues on the surface of the cell, what it will do is it will release chemicals onto the surface of the cell which will break that cell up and kill it, and that will kill that virally infected cell. So all of these lymphocyte responses are helping to kill the virus. And the key thing that we're learning now is that when these B cells and T cells, when the T cells learn to help the B cells and when the T cells learn to produce the cytotoxic specific T cells against viral cells that are infected with SARS coronavirus 2, they also produce memory cells that remember how to do this. So if they're re-exposed to the antigen again, they're not starting from scratch, and they can mount this immunological reaction really quickly. So that's the background science. Now, this study is called Neutralizing Antibodies Correlate with Protection from SARS coronavirus 2 in Humans During a Fishing Vessel Outbreak with a High Attack Rate. So what's happened here is Washington State are following up fishing vessels, and they're testing people before they go to sea. Then there's an outbreak, they can test them after. So it's University of Washington Internal Review Board. So now these are not peer reviewed yet, but the University of Washington wouldn't let out a Mickey Mouse study. So although they're not peer reviewed, it's the first time that this data has become in the public domain. The identification of immunological correlates would help research. So what is it that changes in the body that means that someone is immune? Because so far we only have animal models to demonstrate that. So this was an outbreak of SARS-CoV-2 on a fishing boat. Well, vessel, must have been quite a big boat. Now the attack rate on board was 85.2%. 104 people out of 122 crew became infected during the voyage. So a lot of people got the virus, as you would expect, of course, in a confined space like a fishing boat. Now the pre-departure and serological and viral PCR testing was done. So they tested for the antibody and for antibodies and for the antigen before the boat departed. So they knew what it was before it departed. And no one was testing positive when the boat departed, otherwise, of course, they wouldn't have been allowed on the boat. But someone must have been incubating the virus. And they did repeat testing when they returned to shore. And they got full data on 120 of the 122 crew, 9 women, mostly men. 18-day trip, the fishing trip was reduced because a crew member required hospitalisation from COVID-19, and they followed these up for a medium of 32 days, some up to 50 days. Right, so that's the story so far. So this fishing boat went out. No one had the live virus at the time, but they tested everyone for antibodies and antigen before they went out. 3 people actually had the antibodies before they went out because they'd been previously exposed. Now, what's happening here? 104 individuals had a positive antigen or serological test during the follow-up period. So 104 people caught it, basically, and had evidence of that via the antibody or the antigen test. They'd actually caught this infection that would spread out in the boat. And 101 of the 104 developed immunoglobulin type G, that's the antibodies, within 50 days. Most much quicker than that. So this is showing that the 104 individuals that had been infected, most of them developed antibodies. So these people were susceptible to COVID-19 because most of them hadn't had it. Only 3 had the antibodies out of the 122. 104 caught it during the trip, and of that 104, 101 developed antibodies. Now that in itself is encouraging. And they actually did further studies to test these antibodies and did find out they did work against the virus. So that's encouraging. Now, 3 crew members were seropositive prior to the boat's departure. So they tested the crew before they got on the boat, and 3 of them had the antibodies, meaning that they had the infection sometime in the past. And none of those 3 crew members who had the antibodies developed symptoms or tested positive for the antigen. So even although these 3 crew members who had had the infection before were in this closed environment, and 104 of their shipmates contracted the virus, they did not, nor did they have any symptoms. So that's encouraging. And then when these 3 came back, they took further blood samples from them that they kindly volunteered. And they showed that their antibodies had neutralizing activity, that their antibodies were working. Now, what this usually means, and what it meant in this situation, was, as we know, the SARS coronavirus has got these spikes on it. It's these spikes that bind into the cells to cause the infection. So it's these spikes that are the infective part. So if you can suppress those, that's good. And what happened was these antibodies fitted onto these spikes like this and stopped them from infecting any more human cells. So the antibodies there in red did their job and neutralized. They were neutralizing antibodies. They neutralized the virus. So when the virus comes along to try and infect a cell, there's the receptor site on the cell where it will go into. Normally the viral particle could go in there like that, and the virus could absorb onto that cell and cause infection. But now, of course, it couldn't do that because that big red antibody there was getting in the way, and it prevented the binding into the ACE2 receptor site. So presence of neutralizing antibodies from prior infection was significantly associated with protection against reinfection, and that just means there's a very small chance that result would have risen by chance. It's what is it? So if p equals 0.1, that's a 10% chance. If p equals 0.01, that's 1 in 100. If p equals 0.001, that's 1 in 1,000. So there's only 2 chances in 1,000 that result would have arisen by chance. So it's a pretty significant finding. And as we saw, all those scientists were bouncing up and down with glee at how good this was. So this study shows that most people develop antibodies. The vast majority develop antibodies if they're carefully followed up. That these antibodies work. And they actually, not only did they work in the lab, they work in vitro, but they also work in vivo because none of these 3 became infected, despite being in that high risk environment. So this is the first time that this has actually been demonstrated in humans that this is effective. Now we could leave it there, but I've got 2 other studies that collaborate this. If you can stick with it, this is remarkable. That's a remarkably encouraging study, remarkably encouraging, and I expect that this crew is going to be followed up longer term as well to check on the maintenance of the cellular immunity generated by these lymphocytes. Specific lymphocytes that are made specifically to attack that particular viral infection. Now, this study here, robust T-cell immunity in convalescent individuals with asymptomatic or mild coronavirus disease. Now, this study is absolutely brilliant because it shows that people who've had mild disease or even asymptomatic disease still develop immunity. And up to now, this has been a big question. Is it only people that get really, really sick that develop immunity? Well, no. It's looking now more and more likely that people who even have mild disease or even asymptomatic disease still come to enjoy immunity, therefore will not be infected themselves and also will contribute towards herd immunity. Now, this study was done in Stockholm. Now, this is one heck of a study, this one. There's only a few hundred people in it, but it's remarkably thorough. The analysed groups of sort of 30 and 40 in different clusters. It took me about 2 hours to work out what the heck they were talking about, actually. But once you actually get through it, it's actually a really clever study. So I'm just going to try and give you the bottom lines on it now. And not only is it a clever study, the implications are quite profound. So now this is the first thing. Virus-specific memory T cells. So remember these T cells. We want T cells to remember how to instruct the B cells to make the antibodies, and we want T cells that remember the particular viral part, so they selectively destroy cells invaded with that virus. We need these memory T cells, and they've been shown to persist for many years in SARS coronavirus type 1, 2002-2003 outbreak. So very similar virus. The memory T cells live for years. So we would assume that if someone who was infected with SARS coronavirus 1 in 2003 was reinfected with it, now they can't be because that virus no longer exists, at least not in the wild. So it might exist in some laboratories. I'm not sure about that, actually. I suspect they've got samples of it in these biological warfare laboratories that are maintained in certain countries. In my country, it's at Porton Down. So whether they've got samples or not, I don't know. Someone from Porton Down wants to write in and tell me they can. I think that's unlikely. Anyway, what it shows is that if these people were exposed even now, they'd probably still be immune to that virus. All these years later. Long-lived immunity. Many years. So why should SARS coronavirus 2 be any different? Acute phase SARS coronavirus 2. Specific T cells display high cytotoxic activity. So acute phase means these are the cells that are made when someone's infected. And it's to SARS coronavirus 2. This is the virus that's causing the current pandemic, the COVID-19. So the body makes specific T cells that will attack this virus and not attack other viruses. And they display high cytotoxic activity. In other words, they're very good at cell toxic. They are toxic to it. So these T cells are very good at recognizing the body cells that are infected with SARS coronavirus 2 and will simply kill these cells. They will chuck a hand grenade in and kill the whole cell. Sounds dramatic, but it works. This is how viral infections are one of the main modalities via which viral infections are eradicated from the body. So it's been shown that the T cells to SARS coronavirus 2 are very good at tossing in hand grenades and killing all the viral terrorists inside a particular cell. And they also found this correlates with various clinical markers of disease. In other words, people with good T cell responses had less severe disease. Excellent. SARS coronavirus 2 specific T cells were detected in antibody seronegative family members. So what they did was they took the families that had been infected and they took relatives and they tested the relatives for antibodies. And some of those relatives were seronegative. They did not have the antibodies. They did not have the coronavirus 2 antibodies. They didn't have the SARS-CoV-2 antibodies. But they did have the T cells. And of course the T cells can kill the viral cells and instruct the B cells to make more antibodies should they need it. So even the minority that were seronegative still had protective T cells in family members. And individuals with a history of asymptomatic or mild coronavirus disease. So protective T cells were present in individuals who had asymptomatic or mild coronavirus disease. So what this means is it looks like this long-lived T cell response will be developed in people that are sick. It's very good at killing the infected cells. They have this acute response. But even in people that had very mild disease or were asymptomatic, who'd had the infection asymptomatically as a result of exposure with other family members, they still produced a strong, specific T cell response, meaning they are very, 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 very likely to be immune to any future exposure to the SARS coronavirus 2. So they should not get COVID-19 disease. So this is remarkably encouraging data from Sweden. And as I say, it's a very detailed paper. It took me 2 or 3 hours to work it out. But this is what they're saying. Collective data sets shows that SARS coronavirus 2 elicits robust memory T cell response regardless of the severity of the disease. This is absolutely game-changing knowledge. And the T cell response observed was akin to those observed in the context of successful vaccines, meaning that the T cells that are generated by the vaccines look the same as the T cell response that are generated by having the natural disease, or even just being exposed to the natural virus without developing symptoms, meaning that the vaccine is probably going to be effective as well. It's good news on both of those fronts there. Natural exposure or infection may prevent recurrent episodes of severe coronavirus 2 also in seronegative individuals. So that's what we've said. Even if people haven't got the antibodies now, if they've got the T cells and they're then exposed to infection, they should not get severe disease. They might get these are talking very cautiously. They won't get severe disease. So they're intimating there they might get a mild disease or they might have asymptomatic disease. They're just saying they won't get severe disease. This is written with scientific caution, which of course is good. SARS coronavirus 2 induces robust memory T cell response in antibody seronegative and antibody seropositive individuals with asymptomatic or mild coronavirus disease. So this is saying whether you've got the antibodies or whether you haven't got the antibodies, you still get the immunity from the T cells. Incredibly good news. Now I've put the link to that. Do click on it. If you like scientific stuff and you want to spend about 2 or 3 hours reading it, I think you'll come to the same conclusions that I did. But of all the studies I've reviewed so far making this video, that one was the hardest work. I needed a cup of tea after that one. But that's what it was saying. Now the last one. Functional SARS coronavirus 2 specific immune memory persists after mild COVID-19. So even after mild COVID-19 you get immunity. And this one was from, I think this one was University of Washington as well. Oh, by the way, that fishing boat was Washington State. It was out of Seattle. Now this study, this study was approved by the University of Washington Institutional Review, same as the first one. Recently emerged SARS coronavirus 2 virus is currently causing the pandemic continues to rise. Well, we know that. The majority of infected individuals experience only mildly symptomatic disease. So yes, that's true. Most people get mild disease. but it is unknown whether this can induce persistent immune memory that might contribute to herd immunity. So that was unknown when they published this study. Of course, the two studies we've just looked at intimate that it will produce long-term immunity. But what does this particular study add? Well, they did longitudinal follow-up over 3 months. Individuals recovered from mildly symptomatic COVID-19 disease were studied to determine if they developed sustained immunological memory, these specific B and T cells that could remember exactly how to clobber the SARS coronavirus 2. Recovered individuals developed SARS coronavirus 2 specific immunoglobulin antibody. So even people with milder disease developed the antibody, which was good. The IgG antibody is the longer lived antibody, as you might remember. Also, they developed virus-specific memory B and T cells. So this study also agrees. Now, this study was done on relatively small numbers of people, so it does need duplicated on a much larger scale, but it was done very thoroughly. So I'm certainly buying the results of this study. In some cases, increased numerically over the 3 months following symptom onset. So what this study is saying is not only were they able to identify the activated specific T cells, the memory T cells which would fight the SARS coronavirus 2, preventing COVID-19, they found that in some individuals they studied, the numbers of these protected T cells continued to increase for 3 months after initial infection. So very encouraging. Memory lymphocytes exhibited characteristics associated with potent antiviral activity. So the memory T cells secreted this interferon gamma. Now, interferon is a chemical that goes from one cell to another. So this interferon gamma is released by T cells, goes to other immunological cells and says, "Oi, battle cry, get ready for action." Stimulates the division of the specific cells and does all sorts of immunological priming things. So the memory T cells did that, and they rapidly expanded upon antigen re-encounter. Now, what that means, the antigen, of course, is the virus. So if the virus came back, then the number of these T cells would rapidly divide. So one T cell would quickly divide into two. Each of those would divide into two more. Each of those would divide into two more. And you can see we've got rapid multiplication of these specific activated T cells. This is specific immunity. It's exactly what we want. It's exactly what I suspected we would get. And it's now what this epidemiological work, as well as this in vitro work, is suggesting that we are getting. So if someone's re-exposed, antigen re-encounter, if they're exposed to the virus again next month, they shouldn't get the disease. And the memory B cells, they expressed the specific antibodies that would neutralize the virus. Right. Right. Well, that was good, wasn't it? So 3 different studies there, all showing basically the same thing, indicating that regardless of how severe the COVID-19 was, that long-lived immunity is looking very likely from all of those 3 studies. And the reason I'm particularly encouraged is those 3 studies all agree with each other. So to me, this means that herd immunity will be achievable. We still don't know whether it's at 50, 60, 70%. It means that the vaccines are very likely to be effective because the vaccines are generating these antibodies, and we know that the vaccines are generating this T-cell response. We now know that people with a good T-cell response get less severe disease, and we now know that people with a good T-cell response that lasts for some time afterwards should be immune from re-exposure to SARS coronavirus 2, therefore should not get COVID-19 disease. And to me, this means that we're going to get rid of this virus, partly by herd immunity, but then when the vaccination comes along, I think we can eradicate it altogether and it will be consigned to the history books the same as SARS coronavirus 1. Now, having said that, I haven't got enough information to state that definitively, but that's the way my thinking's been developing for some time. And these are 3 very good pieces of science that indicate that is likely to be the case. If you watch this video all the way through and you followed that, do give yourself some reward, a nice cup of tea or coffee or something. Thank you for watching as always.